the Buchner discovered the enzyme, that is the zymase enzyme is extracted from the yeast cells. So in this alcohol fermentation here, two steps are involved. For the first step, the pyruvic acid is the decarboxylated into acetaldehyde and the carbon dioxide. In the presence of pyruvic acid, the hydrogenase. Okay, so in the next step, these acetaldehyde and again reduced to the ethyl alcohol by taking the NADH and to produce the alcohol, that is the with the help of the alcohol dehydrogenase enzyme. So finally what happened here, the glucose means pyruvic acid then converted into alcohol and carbon dioxide and NAD. The next one is the lactic acid fermentation. So next one is the lactic acid fermentation here, which is followed the lactic acid bacillus. So here the some bacillus, lactic acid bacillus is involved for the fermentation of the lactic acid. So in this process, the lactic acid is instead of the alcohol and carbon dioxide are formed. In this process, the best known for it is the source of storing the effect on milk. The pyruvic acid molecule found in the glycolysis, which are directly reduced by the NADH to lactic acid. So here the main enzyme is the lactic acid dehydrogenase is involved. Okay, so pyruvic acid directly reduced to the lactic acid with help of the lactic acid dehydrogenase. So here, okay. The next one we discuss the nitrogen fixation. So nitrogen fixation. So the nitrogen fixation. So the nitrogen is one of the most important element in the all the organisms. We know that one. It is the main constant to the proteins, amino acids, enzymes, nucleic acids, chlorophyll, and other alkaloids also. All the biochemical compounds are made with the nitrogen. Okay. So in our atmosphere, the 78% of the molecule is the nitrogen, but this nitrogen cannot be replaced by plants. So after it is to convert into the nitrite and nitrates and ammonia by with help of the nitrogenase enzyme. తీసుకొని పోవాలి. So which is only the prokaryotic process this possesses this enzyme. Okay. So here, so these plants absorb the nitrogen from the soil mainly in the form of nitrite and ammonia, but the nitrogen gas cannot be replaced by the plant as directly. Okay, so this nitrate reduction takes place in the root as well as some leaves also. The reduction of the nitrate is the fundamental physiological process. The oxidized from the nitrogen is converted to ammonia. Here mainly two enzymes are involved in the conversion of the nitrogen to the nitrates. and ammonia. So here main two enzymes are there, that is the nitrate reductase and nitrate reductase. These two enzymes are mainly involved. The main what happened here, the nitrates are reduced or reluctant to form the nitrate in the presence of nitrate reductase. So after nitrate, again to reduce the ammonia, the presence of nitrate reductase. So these nitrate reductase is common enzyme found in the prokaryotes and eukaryotes also. So this is important enzymes. These two enzymes are so involved in the formation of the or reduce the ammonia. So this nitrate reductase is the monomeric polypeptide enzyme is there. So here the pyridoxine, the main element in the nitrate reductase. Okay, so after the nitrogen fixation, how the fix the nitrogen into the soil as a biological or okay. So here you can see this one here. Then atmospheric nitrogen to fix the so the nitrogen fixation. The nitrogen is one of the already we discussed that one is the essential nutrient for the growth of the plants. Okay. The nitrogen is in the form of nitrate and ammonia. They are available in the soil. They are utilized by the plants. So here, this nitrogen fixed by the in chemically, physiologically, as well as the biologically. Okay. So in these three methods are the atmospheric nitrogen to fix in the soil in the form of the ammonia and nitrates. So as the industrial fixation means here, a wide variety of nitrogen chemicals are manufactured. So the chemicals are manufactured in the industry. The chemical method. So here, this is the fix. They prepare the manufacture the some chemicals. The ammonia chemicals are prepared in the industrial area. So next one is the physical nitrogen fixation. So in this physical nitrogen fixation, about only 10% is the to fix the atmospheric nitrogen in the process of physical process such as the lighting strikes and thunderstorms. So these two methods, the lighting sticks and thunderstorms, there are the lighting is dissociated with the water vapor into the hydrogen and oxygen atom and hydroxyl radicals is also. So these free atoms and radicals which are exchanged to combine with the nitrogen in the presence of atmosphere and produce the fixed nitrogen gas. So these gases, nitrogen and oxygen, combine to form the nitric acid. So nitric oxide is formed. So this nitric oxide is further oxidized to nitrogen peroxide. So this nitrogen peroxide is combined with the water during the rainy and to produce the nitric acid. So here the photochemical reactions between the ozone and nitrogen dioxide. So this produced to combine with the alkaline radicals to produce the nitrates and nitrates during the rainy season. So another fixation is the biological fixation. Another method is the biological fixation. So the transfer of the free insert nitrogen of the atmosphere into the fixed form by the soil with help of the microorganism, which is known as the biological fixation. Means here some microorganisms are involved to fix the atmospheric nitrogen into the nitrates and nitrates. So this is the biological nitrogen fixation. So here nitrogen fixation in the soil is carried by the both free living, there is the symbiotic and symbiotic microorganism. They are called as the diazotrope. So here asymbiotic, similarly asymbiotic nitrogen fixation is also there. That is the aerobic, that is the Azotobacter and Klebsiella azospirillum. These are the asymbiotic bacteria to fix the atmospheric nitrogen to the soil. So another one is the anaerobic nitrogen fixation, that is the Clostridium and Rhodospirillum thiobacillus. These three bacteria are the anaerobic bacteria. There is the non-photosynthetic bacteria is the Clostridium. Photosynthetic bacteria is the Rhodospirillum. Another one is the chemoautotrophs, so that is the thiobacillus. This is the, so another is the cyanobacteria is also fix the nitrogen. So some filamentous forms are there, that is the heterocyst, with help of the heterocyst that fix the nitrogen, and unicellular forms also there. So unicellular forms are the glaucocarpa, glaucocapta. Another one is the filamentous. Filamentous heterocysts also there, that is the nostoc and anabina. Non-heterocyst forms also fix the nitrogen. Example, the Acidoria and Plectonium. So another one is the symbiotic fixation. So here symbiotic fixation is the legume symbiosis and non-legume symbiosis also there. So legume symbiosis means the rhizobium, legume plants. So the legume root nodules are associated with the rhizobium bacteria to fix the nitrogen into the nitrates and nitrates. So legume plants, example, the Phaseolus. Okay, Phaseolus and Pisces sativa. These two are the rhizobium bacteria also there. So non-legume symbiosis is also there. That is the ferns and the cycas and lichens. Okay. So ferns also azolla. Azolla belongs to the fern. This azolla is associated with the anabina. Some cycas corallide roots also. This is associated with the anabina and nostoc. For example, lichens is also. So lichens is associated with the nostoc. So some fungi and algae, they are to combine together to form the lichens. These lichens also fix the nitrogen into the ammonia. So these some blue-green algae. So blue-green algae, for example, the Nasticaceae family and Cytonomiaceae, Rivulariaceae and Stigomataceae family. They are fixed the nitrogen.
Excuse me, sir.
Hello?
Hello? Can you
Sorry?
share the screen once again?
Hello?
Sir, please share the screen once again. We are unable to see what you are saying.
Okay. So here the symbiotic fixation means here nitrogenous complex. We discuss this one. Is clear this this picture?
Yes, sir.
Okay. So we
Okay.
discussed the nitrogen is the enzyme. Nitrogen is enzyme mainly to involve the ammonia, means that nitrogen into the ammonia. Okay. So here the nitrogen fixation in the different methods are there. Different methods means there are the chemical fixation and physiological and biochemical fixation are there. Okay, biological fixation. So mainly we concentrate the biological fixation, symbiotic nitrogen fixation. In this symbiotic nitrogen fixation means the nodule formation. Nodules are formed, root nodules. Okay, is clear? Hello? Okay, Nadi.
Sir,
Root nodule formation. Okay. We discuss later, but here the cyanobacteria. Okay. Here blue-green algae. These blue-green algae members to fix the nitrogen. So with help of the heterocyst, the Oscillatoria and nostoc, this anabina, these are the fix the nitrogen. So another one is the asymbiotic bacteria also, for example, Azotobacter and Beijering. These are the some important aerobic bacteria. So there is no symbiotic. There are these bacteria directly fix the atmospheric nitrogen. And another one is the azospirillum is also. Azospirillum, Clostridium. This is the asymbiotic nitrogen bacteria. They are directly fix the nitrogen. So another one is the symbiotic nitrogen fixation. So here the symbiotic nitrogen fixation, here nodule formation and the non-legume, some legume plants and non-legume plants also involved in the formation of the nodule. So here the legume rhizobium symbiosis, we discussed that one. The legume plants and rhizobium bacteria, between these association is the symbiotic association. So here first of all, the initially the infections of the roots are formed on the root nodules. So here the root hairs are to form a cluster. Root hairs are formed. So these infected nodules, infected root hairs. Okay. So here infected root hairs are to identified by the rhizobia, rhizobia bacteria. So that roots are the root hairs. Okay, root hairs are to produce some flavonoids and lectins. These are the carbohydrate compounds. So these flavonoids and lectins are to attract the rhizobia. So rhizobia, you know that rhizobia is the plenty of the rhizobia present in the soil. Okay. So these flavonoids and lectins are to attract the rhizobia. They are to gather in the around the root hairs. So in the response of bacteria, root hairs to curl at their root tips. Root tips are to modified as a curl. So the bacteria then invade into the root hairs. The plant is produced the infection thread. Means after entering to the bacteria into the root hairs, so gradually the root hair are curved and it is modified as the infection thread. A thread-like structures are there. There are two infoldings. They are formed. Infoldings are there. So these rhizobium signal molecule to play a key role to induction of the nodulation. That is known as a nodule factor. Okay. So here there are the nodules. The bacteria is directly enter into the endodermis and cortex cells. So that is connect with the vascular tissues. The bacteria directly connect with the vascular tissues also. So during the nodule development, the bacteria are transmitted to the bacteroids, which are several times larger than the bacteria. So the transformation of the bacteria to bacteria is closely related to the synthesis of leg hemoglobin. Leg hemoglobin and nitrogenase and other molecules required for the nitrogen fixation. So this is the that leg hemoglobin and nitrogenase enzyme. Leg hemoglobin is the pink in color compound, which is the uh, very essential in the fixation of the nitrogen. Okay, so these bacteroids are the to observe the nitrogenase into the from the soil from the atmosphere and fix the nitrates and nitrates into the soil. So here some ATP is also utilized by the plants. So here means they are to take the shelter from the legume plant. The bacteria are taking the shelter from the legume roots. So that's why this is a symbiotic relation is there between the legumes and rhizobia bacteria. Okay. So the next one is the so already we discussed that one. So another one is the asymbiotic relation. So asymbiotic relation means there is no any relation means there is the some bacteria, free living bacteria are there. There is Azotobacter and Clostridium. We already previously discussed that one. Some filamentous forms and blue-green algae members are also there. So here mainly what happened here, the nitrogen cycle, we discussed that one. How to fix the nitrogen from the atmosphere to the soil. So what happened here, the nitrogen is symbiotic, means symbiotically or asymbiotically to fix the nitrogen. So in the form of the amino acids and proteins and that is the nitrogen compounds means ammonia and nitrates and nitrates. So these ammonia compounds means nitrogenous compounds are utilized by the higher plants. So they are directly to observe the plant. So after they are to destroyed in the, some fungi. So convert into the organic compounds or and inorganic compounds are there. So which are result to the ammonia and nitrate. So here there are two processes are there, two distinct microbial processes are there. That is the mineralization and formation of ammonia known as the ammonification. After destroying the leaves and stems and animal parts, that is what happened here. They are destroyed. So that is the decomposition means that is the mineralization. So that is formed in the ammonia that is known as the ammonification. So ammonifying bacteria is also involved in this process. So the ammonification of the soil microorganism converts into ammonia, nitrogen to ammonia. The ammonia reacts with the carbon dioxide and water present in the soil, form the ammonium salts. So in the nitrification, the chematotrophic bacteria, that is the nitrosomonas obtain the energy by oxidizing the ammonia in the form of ammonium salt to nitrate. So after nitrification, what happened here? The denitrifying bacteria. So these thiobacillus denitrifying bacteria, that is oxidize the sulfur to produce the nitrate to nitrogen. Again, they are the denitrification. That is the thiobacillus. The nitrate again to convert to the nitrogen and to release it to the atmosphere. So this is the cycle is involved in the nitrogen fixation, biological nitrogen cycle. So mainly here biologically involved. That's why we discuss the biological nitrogen fixation. Okay, Nandi. So next one we discuss the some plant regulators. That is the plant promoters and plant hormones. Discuss that. Okay. Plant growth hormones and plant regulator hormones. Plant regulators. So the growth is defined as a gradual increase in the size. You know that in a plant or a tree, that is the gradually it is increased. So that is the main characteristic feature of the plants. So there is the in the length and mass. That means growth is the volume or in the weight. That is nothing but the growth. So here we discuss the plant growth is generally restricted to some regions that is known as the meristematic region. In plant body, a particular region is there, that is the meristematic region that we made up of the meristematic tissue. So this meristematic tissue is present in the apical region and intercalary cells also there. So the apical region they are to divide very rapidly to produce the cell elongation, means the plant growth, and intercalary cells that is rapidly grow to develop as the weight, means radiationally they are to produce. So here growth we are considered the three phases are there in the plant growth. The meristematic tissues to produce the cells in the three phases are there. So first one is these three phases is called as a SAT graph period is also discussed that one. Growth curve. Sachs growth curve we also call Sachs growth. So here first of all the cell division phase, next one is the cell elongation phase and maturation stage. Okay. Cell division phase and cell elongation and maturation phase. So the cell division phase means the first phase is what happened here. The slowly it is present the cell division phase, that is the log phase. So slowly it is developed. That one we call as the log phase. After some time, the rapidly elongation is present the phase of the cell elongation phase, that is the log phase. And finally, that is the saturated phase, cell maturation phase, means at the end of the cell division. So it is they developed the Sachs, that one, the Sachs curve is there. Okay. So these plant regulators, that is the hormones are there, that is the natural and synthetic hormones are there. There are the Five types of the plant hormones are there. So out of the five types, three are means include the auxins, gibberellins, and cytokines. These three are the plant growth hormones. Out of five means auxins and gibberellins and cytokines. These three hormones are the plant growth hormones. And another ethylene and abscisic acid. Ethylene and abscisic acid. These are the plant inhibitory hormones. These two are called as the plant inhibitory hormone. Okay, so first of all, discuss the auxins. Okay, so the plant's growth or development under the two sets of the internal factors are there. That we know that one. That is the nutritional factor. Another one the and some raw material is requirement are there. Because some nutrition is also essential for the development of the plant. So these plant hormones are involved in the increase in size. So they are called as growth hormones and growth substances, growth regulators, and growth factors also. Okay. So here, growth regulators means the plant growth regulators. So they naturally or they are some synthesized. So IAA, indoleacetic acid. These axons are naturally they are produced as well as synthesized also. Indole butyric acid, IBA, these axons is always synthesized, not naturally. So the plant hormone, there is some important features are there. Their physiologically and chemical response. There is these hormones is the organic nature is there. So naturally the occurrences, and the third character is the mobility within the plant and activity when the present is extremely low concentration. And another character is the influence in the growth and development. These five features are essential in the plant growth hormone and plant promoter. So what is that one here? These hormones are organic compounds. They are naturally occur and they are easily to mobility within the plant. So very active at the low concentration, and which is the influence in the growth and development. These five characters are shows in the hormone. So first we discuss the auxins. The auxins. Okay. So auxins is derived from the Greek word that is means grow or increase. So the first plant hormones is discovered as the auxin. So first of all, the Sachs was the first person that is the organ forming the substances, the producing the leaves and translate downwards. So after Charles Darwin observed that the young seedlings are canary grasses elongate the curve toward the source of light. By using the light proof cap that is able to show the light sensitivity region was observed in the apical tip of the seedlings. And not region further backs which are actually underworked in bending. So that is the what happened here. They grow into positive way towards the sunlight, means light focusing area. So after some days WF went. So he cut a coleoptile and placed on them the agar block for 1 hour. When this agar block was replaced on the stump and curvature occurred, that shows the substance from tip to downwards into the agar block. So after he is to employed for the extraction of the some hormone, that is the indolestic acid. So as the oxygen is soluble in the water, alcohol, chloroform. So this is extracted by the solvent method. Uh, so the distribution. So the distribution of the oxygens in higher plant, a survey of the oxygens is petuolated oat seedlings has shown that the greatest quantities occur in the roots and shoot apex. Very essential in the root and shoots. So what are the some applications of the oxygen? Occurrence and distribution of the oxygen. What is the occurrence? The occurrence is usually in the plant, whereas the very active growth wherein the oxygens are produced. So active growth means shoot apex and root apex. So apical body synthesis the more oxygen than the lateral buds. So that is the apical dormancy, that one. So the apical buds are very actively synthesized than the lateral buds. Okay. So next one is the effect on the different organs in plant growth. So first one is the cell elongation and cell division. Cell elongation and cell division. So these organs cause the coleoptile and stem due to the elongation of the existing cell. So it is increasing the osmotic content and permeability of the cell through water and wall synthesis. So it is by the reducing the wall pressure. Okay. So organs also induce the promote the cell division with the cambial region is also. Okay. So here cell elongation is the main function in the oxygen. The next one is the apical dormancy. So in some plants is terminal bud is the dominant and inhibit the growth of the lateral bud. So this phenomenon is called the bud inhibitory. It is generally the with help of the oxygen. So what happened here? The apical buds only gradually develop than the lateral buds. So this phenomenon is apical. So what happened here? So we have to cut into that apical bud and keep the a potato tuber or any other one. So that is the lateral buds is also growth. And next one is the rooting. Next application is the rooting. So the stems are cut when applied in the some auxins So the auxins are to promote the root formation. So here IBA, indole acid, is used for the rooting. So it is the commercially applied. So next one is the parthenocarpy. So next one is the parthenocarpy. The process in the development of the fruit without fertilization is called the parthenocarpy. So without fertilization, the fruits are formed. The process is called as a parthenocarpy. So the development of fruits in the ovaries of flowers and possible and develop the ovules into mature seeds after fertilization. They are controlled in the part of oxygen. So the fertilization is controlled by the oxygen. So that's why the seeds are not developed. So the oxygens also to introduce the parthenocarpy. And next one is the growth movement. The plants may react with the growth movement in the directional stimuli, particularly the light and gravity movement. So such movement termed as the tropical movement. The tropic movement of the means they are include the geotropism and phototropism. Since the plants are stimulated to the light and gravity. So to react with the plant light, that is the phototropism, and react with the gravity, that is the geotropism. So here the geotropism, the movement of the plant organs in response to the force of the gravity is called the geotropism. So this is response to the force of the gravity is called geotropism. Whereas the stems grows away from the center of gravity and known as the negative geotropism. The roots and branches and leaves are some places the horizontal and they are to develop. So in the lateral roots are the not since the gravity, there are the azotrophs. Gravity is not a receive through the whole plant and perceptive region appears to the both root cap. Okay. So next one is the phototropism. So the phototropism, the plant then bends to the light towards the light. So this is to the higher concentration of oxygens on the shaded side. So what happened here on the shaded side, the oxygens are to rapidly grow. So that's why the plants towards the light side. The movement of the plant organ as determined by the direction of the incidence of the rays is called phototropism. So the stem is towards the source of light and known as the positive phototropism is there. Okay. So the next one is the so geotropism is already discussed that one. The next application is the routing and control the abscission. So prevention of the abscission. Okay. So the presence of oxygen, the presence of oxygens, high concentration, they are to does not occur in the abscess. So we already discussed the parthenocarpy. Parthenocarpy means fruits without fertilization. Fruits are developed without fertilization. That is another application in the So another one is the callus formation. Callus formation is also in the main organs are developed. The next one is the sex expressions. Auxins introduce the changing the sex ratio of the flowers toward the femaleness. So, so that's why to increase the female flowers. Okay. So next one is the promoting the flowers, promoting the flowering to increase the flowering. So that is the naphtalic NAA, naphthalic acetic acid, and 2,4-D is also used for increase the femaleness in monoecious, that is cucurbitaceae families. So increase the germination capacities also. IAA and IBA, indole acid and indole butyric acid, is mostly used in the soaking seeds for the germination. Okay, so the auxins are widely used in the horticulture and other areas. Okay, so next one is the fruit setting, thinning of the flowers and fruit and leaves, and prevention of the loading in the cereals. Okay, prevention of the lodging in the cereals are different applications are there. So next one is the gibberellic acid. So in the tissue culture we also used, and herbicides is also used as auxins. Okay, so next one is the gibberellic. So auxins are widely used in the herbicide. Okay, next one is the gibberellins. So the gibberellins is strongly promote the plant growth, especially for the stem. So these gibberellins are first to identify the Kurosawa from the Gibberella fusiculae fungi. Okay. So from the Gibberella fusiculae fungi is isolated these gibberellins. So this is the some the Gibberella fusiculae is to cause a Botany disease. Botany disease means there is no flowering and fruiting plants. These Gibberella fusiculae fungi to develop this disease. Okay. So from physiological properties are there. What is that one? Physiological functions means application of the gibberellins. So here stimulate the cell division. Stimulate the cell division. Gibberellin is stimulate the cell division. So cell elongation and control the enzymes and secretion. For drop cultivation can be treated with the gibberellin and grows to normal height to indicate the drop lock normal leaves of the gibberellin. So next one is the involved in the overcoming the dormancy of the seeds and buds, seed dormancy and bud dormancy. What happened here with help of the gibberellins to break the dormancy. So the gibberella is transferred easily to the plant in the both directions, because to produce in the only shoot apex, but also in the root structure is also. So Next application is bolting and flowering. In the rosette plants such as the mustard and cabbage and radish, these leaves developed in the purpose and internodes along in the retard. So that's why we are to apply the gibberellin in these plants, that is the mustard and cabbage plants. What happened here to increase in the stem growth, that known as the sudden increase. Okay, sudden increase the stem growth, that is known as plotting. And seed germination is also. The gibberellins are regarded the germination, promote the germination is also. Another one is the parthenocarpy. Gibberellins is also involved in the parthenocarpy. Okay. And another one is the sex determination is also. Sex determination. So here which are the promote the male flowering in cucurbitaceae family. Okay, so these applications are involved in the gibberellins. So next one is the cytokinin. So these cytokinins, so this also promote the cell division. So cytokinins are promote the cell division. So cell elongation, cell division, cell elongation, and morphogenesis and organ formation. These are the. So cell elongation means cytokinins are mainly concentrated with the cell division. They are also capable for enhancing the cell elongation, particularly in the leaves and cotyledons. And morphogenesis also. So these cytokinins, by interacting with the oxygens, regulate the process of morphogenesis in the plants. That is the what happened here. The cytokinins and oxygens in the different ratio to form the root formation. So next one is the delay in the senescence. So the aging process of the plants is known as the senescence. So older leaves become turned into yellow in color. Older leaves are yellow in color. That gradually what happened here due to the breakdown of the chlorophyll and nucleic acid proteins. They are to detach from the plants. So that senescence breaks with help of the cytokines. So another one is stomatal opening is also. Stomatal opening is also mainly involved in the cytokine. So another one is the another phytohormone is the abscisic acid. Another one is ethylene. So ethylene is a fruiting hormone, which is known as the fruiting hormone. So this is the plant inhibitor. To stimulate the flower has been known as many years. So certain this is the gaseous. Ethylene is the gaseous hormone to introduce the flower. So mainly the ethylene, the simple carbohydrate for the formula that is the C2 and H4. So ethylene is frequently produced when the high concentration of the oxygens are supplied to the plant tissue, which is stimulate the flowering in pineapple and other some plants means that is the okay. So to wound healing is also another application is the wound healing is there to with help of ethylene, the cell division and help to process the killing of the wound in the plant. So increase the root to hair formation is also increase the production of the pistil means female flowers and reduce the apical dormancy to reduce the apical dormancy. Okay, so these are the helpful in the ethylene. Ethylene. So next one is the abscisic acid. Next plant inhibitory hormone, that is the abscisic acid. That is the abscisic acid. Hmm? So abscisic acid, this is the resistance the dot. So which is the growth inhibitory hormone, which is the fighter hormone. This is called as a drought resistance hormone. Drought resistance hormone called as that one. So the physical process are there, that is the dormancy to develop the dormancy. In the earlier study, we attempt to establish between the abscisic acid concentration and dormancy. That is the abscisic acid to increase the dormancy. So stomatal movement is also another application is the stomatal movement. So the abscisic acid to increase the close the stomata. So it is induces the stomatal closing. So that's why to lose the water due to the transpiration. So in this, the act as a natural anti-transpirant plant, which helps the conversion of the water during the drought stress condition. The abscisic acid is very helpful in the drought condition. And another one is the seed germination. So seed germination during the maturation of the embryo. So abscisic acid is accumulated in the large quantities